Good evening, everyone. So, this week we'll be starting a discussion on animal models in leprosy. Now, this topic is a bit of dry topic, but it's a very important topic from theoretical point of view. That means in exams you might get a short note on animal models in leprosy or the animal models that are used for research in leprosy. Okay, so for a few more weeks now, we'll be discussing topics from leprosy, about 4 to 5 video lectures, and the purpose of this video lecture is to get you accustomed about the different animal models which are used to research in leprosy. Remember, leprosy is a unique infection in that it takes a lot of time to develop. There's a long incubation period, and it infects humans, and it is so variable in its clinical outcomes that we need to study more about the disease so that we can have good, uh, you know, good diagnostic and therapeutic modalities to help us fight the infection. Okay. Since leprosy requires an animal tissue to grow, it's difficult to grow without an animal tissue. So we require animal models so that research can happen properly in leprosy. Clear? Without much further ado, let's start a discussion on animal models in leprosy. Let's move forward. Now, leprosy is one of the earliest correlation of microorganisms causing human disease. That means it was established multiple years back, decades and centuries back, that M. leprae causes leprosy. In fact, if you know the name of the scientist who correlated M. leprae to leprosy, mention that in the comment section below. Okay. Now, M. leprae causes leprosy, but there is no way to cultivate M. leprae outside the tissue or outside a living organism. Clear? Now, most of the bacteria you can culture in culture plates, but culturing leprosy becomes difficult because you require active living animal tissue to grow. That's why the research is deficient. That's why we are not able to properly study leprosy because we don't have good animal models. Okay. The organism M. leprae requires living tissue to grow, and that is why we require animal models. Also, the initial stages of infection is missed. Remember, leprosy has a very long incubation period. A person might get infected and then develop the disease after like multiple years. After many years, the disease becomes clinically apparent and you can diagnose leprosy. So, with that in mind, we should know that what happens in the initial stages of infection. So, we need an animal model where we can infect or inoculate with M. leprae and see what happens in the initial stages of infection. Clear? Now, three scientists that you should remember is one is Charles Shepherd. Charles Shepherd was responsible for inoculating foot pads of mice. Okay, so mice was used by Charles Shepherd, and then mice was the initial organism of choice for M. leprae. But eventually, Kirkheimer and Stoss were two scientists who were able to develop Ellen Hansen in nine-banded armadillo. And after that, we have been using nine-banded armadillo as animal model of choice. Okay, as animal model of choice to study leprosy infection. Okay, so three scientists, Charles Shepherd for mice and Kirkheimer and Storrs for nine-banded armadillo. Clear? Let's move forward. So animal models can be divided into two parts. Either you have murine models, which are the mouse, and the non-human primate models or apes and monkeys. So if you get a short note on animal models in leprosy, you need to mention these two lists, murine models and non-human primate models. In murine models, you have immunocompetent mice in which the immunity is normal, or immunosuppressed mice in which the immunity is suppressed artificially so that we can look at what happens when the M. leprae infects an organism where immunity is suppressed. What do we mean by that? Now, M. leprae, we know that leprosy has a clinical spectrum. You have the tuberculoid pole in which the immunity is strong and the bacilli M. leprae is sequestered and controlled. While on the other hand, you have the lepromatous pole where the immunity is low and there's a widespread infection. Clear? So we need to have animal models in which we can study the widespread infection also, or we can study the granuloma formation in the tuberculoid pole also. So respectively, we have the immunosuppressed mice for lepromatous pole and immunocompetent mice for the tuberculoid pole. Examples of immunosuppressed mice are T900R mice, nude mice, beige mice, skid mice, and neonatally thymectomized Lewis rats, also known as NTLRs. Okay, coming to non-human primate models, we have the armadillo. Remember, armadillo is the most important animal model of leprosy that we use. Clear? Other and non-human primate models are chimpanzee, mangame monkeys, rhesus monkeys, cynomolgus monkeys, white-handed gibbon, and African green monkey. So remember these names, okay? Let's move forward. Now coming to mouse models, mouse or mus musculus, which is the scientific name. See, why mice models? Mouse models were readily available organisms, readily available animals in labs. So they were used by Charles Shepherd first, and eventually multiple people also use mouse footpad inoculation. In fact, it is the gold standard of animal research before nine-banded armadillo. Okay. Now mice are small, inexpensive mammals. They can be easily housed and they're easy to maintain because of their small size. They are prolific breeders. That means one mice can lead to multiple different mice offsprings. So there is no shortage of animals. There's a short gestation time, so multiple pregnancies can happen one after the other. Two methods are used: foot pad injection and intraneural injections. Okay, either you inject the foot pad or you inject around the nerves. Clear? Let's move forward. So let's start our discussion with the mouse foot pad injection. We know that Charles Shepherd in 1960 used immunocompetent Carworth farm white mice. You need not remember the name, just remember that Charles Shepherd in 1960 was the first to inoculate mouse foot pad for research in leprosy. The advantages of mouse foot pad is that it has a cool temperature. Remember, M. leprae requires cooler sites to grow. It prefers cooler sites. That's why you get slit skin smear done from the earlobe. Earlobes are away from the body core, so the temperature is low. Also, the mice were initially used for research in other mycobacteria like Mycobacteria tuberculosis. So they decided that it's better to use the same animal for M. leprae also. So it was found that when the inoculation size is less than 10 to the power 4, the M. leprae divides as it should and has a generation time of 13 days. Generation time means the time taken for M. leprae to divide from one M. leprae to become two M. leprae. Okay. Generation time in humans is roughly about 10 to 13 days. So when the inoculation size is less than 10 to the power 4, the M. leprae divides normally as it divides in humans. Okay. If the inoculation size is more than 10 to the power 6, the host immunity successfully destroys the bacilli. Okay, it successfully kills the bacilli. In fact, so much so that immunization happens. The host is immunized against the bacilli. So whenever you're inoculating mouse foot pad, remember the inoculation size should be less than 10 to the power 4. It takes roughly about 20 to 24 weeks to become significant. That means after inoculation, after injecting the mouse, it takes about 20 to 24 weeks for the lesion to become apparent. And then you wait for months to 1 year when the lesion is properly developed. And then you sacrifice the mice and you take samples from the mouse footpad and do your research. Clear? Let's move forward. Now, why intraneural injection? In the previous slide, we have discussed about the foot pad inoculation. Why do we need to inject inside or around the nerve? The mouse nerve, the murine nerve, resists natural infection. So we know that in humans, after the infection of skin, the M. leprae is migrating towards the neural tissue and infecting Schwann cells. Okay, mice Schwann cells resist infection by M. leprae, so you might not be able to get good nerve samples for your study. Okay, with that in mind, you might have to inject it around the nerves so as to artificially inoculate M. leprae inside the nerve and then allow it to progress and do your research in the nerve involvement in leprosy. Clear? About 10 to 20 into 10 raised to power 6 is injected into the sciatic nerve. Okay. After that injection, they start to multiply. The M. leprae starts to multiply. We know that nerves of mice have a warm temperature, and we know that M. leprae requires a cooler temperature, and because of that, the multiplication is restricted. So that is why inoculating and multiplying M. leprae in the nerve tissue of mice is difficult. There are certain mice in which we use for intraneural infection, like sensitized Swiss white mice, which is used to study the tubercular type granulomatous response. while unsensitized Swiss white mice are used to learn about macrophage type response. Okay, some mice models do show demyelinations, but we know that in mice they are not a good model to study nerve involvement because they resist the infection. So we require special models to look for the nerve involvement in leprosy. Clear? Let's move forward. So this in this table we'll summarize very quickly about different immunocompromised or immunosuppressed mice models for leprosy. You have the nude mice. Why it is known as nude? Because of a genetic mutation that the scientists have created in the mice. The mice is not able to develop a good immune response, and also it sheds its fur and appears nude. Or not appears nude, is nude. So that is nude mice. The characteristic is that you can use a higher inoculation. Remember, you have to use less than 10 raised to the power 4. If you use more than that, the immunity takes over and the organism, the mice, becomes immunized. But in nude mice, you can use as high as 10 raised to the power 10 bacilli per foot pad. Also, neural invasion is seen, and you can study the nerve involvement also. These two are the advantages of nude mice. Nude mice is used for chemotherapeutic studies, that means to study drugs, mechanism of reversal reactions, and also nasal transmission, which is the important route of transmission in humans. The T900R mice has advantages such that the growth continues after second year, and it can reach up to 10 raised to power 7 to 10 raised to power 8 bacilli. Now remember, in normal mice models, about 10 raised to power 6 bacilli is reached. But in T900R, it can reach as high as 10 raised to power 7. That means 100 to 1000 times, 10 to 100 times the normal bacilli, because the growth continues. The stationary phase is delayed. Okay. Beige mice is new in leprosy studies. Beige mice lacks natural killer cells, and they're used for chemotherapeutic studies. Skid mice, they have an enzymatic defect, and because of that enzymatic defect there is lack of function of T and B lymphocytes. They are used to study persisting infections or persisters, presence of persisters in leprosy. NTLR mice have better survival rate but variable immune response. Clear? So now we have discussed in a single table what are the different mice models, immunocompromised mice models, and what are their advantages or disadvantages. Clear? Let's move forward. Now, how to apply mice models? If you have mice models, what we can do to learn more about leprosy? We can study the granuloma. We can enumerate viability assays. That means we can look at the viability of M. leprae, whether the M. leprae is active or not, drug testing, evaluation of vaccine candidates, and also diagnosis and insights into antigen responsiveness. That means we can study the granulomatous response, we can study whether the M. leprae can cause disease or not, we can study different drugs for therapy of M. leprae, we can look at the efficacy of vaccines and other things that which can use to inoculate against M. leprae, or we can look at what is the antigen response after infection and study more about what actually happens when M. leprae enters the body. Clear? So let's discuss in little detail about each of these applications. Now, if we are using immunocompetent mice, that means mice in which the immunity is okay and it will be able to restrict the infection, what all we can study? We can study because there is limited and minimal growth of M. leprae because immunity is strong. We can study macrophage response and the formation of epithelial cell granulomas, which we see in the tuberculoid poles. We can also study how does the macrophage becomes activated. We can look at the lymphocyte infiltrate. After the inoculation of mouse foot pad in immunocompetent mice, we see that the infiltrate is largely composed of lymphocytes. They are rarely neutrophils, and there's a very rare neural involvement, somewhat similar to what we see in the tuberculoid pole in human infections. Clear? We have bacilli inside the macrophages where they are killed by the host response. So because why they're killed? Because the mice that we're using is immunocompetent. The immunity is able to destroy the infection. Clear? Let's move forward. Now, if we are using immunosuppressed mice in which we have artificially decreased the immunity so that we can look at the widespread M. leprae infection, we use the earthymic mice, which is a type of immunosuppressed mice. The disease mimics LL Hansen. Why? Because immunity is low, widespread infection. Macrophages form leproma, which are heavily infected foamy macrophages replacing normal tissue, like we see in human infection. We have multiple M. leprae. We have M. leprae infecting even the striate muscle cells around the nerves, also Schwann cells and fibroblasts. So neural involvement is seen in immunosuppressed mice. So neural involvement is present. After 1 year, bacilli is seen in virtually all tissues except central nervous system. Clear? Let's move forward. Now, how do we study granuloma? We inoculate the mouse foot pad, we allow the disease to develop, and then we take samples from the foot pad. Clear? There are two methods. There are multiple methods. One is the Shepherd mouse foot pad assay, in which less than 10 to the power 4 highly viable M. leprae are inoculated in the hind foot pad. That means the back foot pads. Okay. This allows the growth to be monitored and the development of granuloma, and they requires about 18 months for the granuloma to properly develop fully. Okay. The other protocol is high dose protocol. That is based on the lepromin test in humans. High dose protocol, you use the inoculation more than 10 to the power 6, and we know that mice models like T900R are used for high dose protocols. They allow for better exploration for good composition because when the inoculation size becomes high, a lot more multiplication can happen and you have a high reservoir of bacilli in the animal. Tissue can be harvested either early, in midway, or more than 32 weeks, as in late. And those tissue samples can then be examined by RT-PCR for different cytokine and chemokine expressions. Other things that you can use is flow cytometry or ELISA, but remember these are just modalities which are used after the mouse foot pad inoculation. We can either inoculate by the Shepherd assay, in which less than 10 to the power 4 bacilli are used, or high dose protocol, in which more than 10 to the power 6 bacilli are used. Clear? Let's move forward. The second application mouse foot pad is the viability assays. That means to find out whether the M. leprae which you have taken from the patient can actually cause the disease. So in some instances, you can take biopsy sample from the human patient and then use that biopsy sample to take out M. leprae and inoculate that M. leprae in the mouse foot pad. Clear? So after inoculation in the mouse foot pad, you allow the M. leprae to grow, take the sample from the mouse foot pad and analyze whether the bacilli can cause the disease or not. Clear? So after taking the sample from the mouse foot pad, either you can stain it for acid-fast bacilli, that is a routine method, and look at the organism. This method is older, depends on whosoever is recording their readings, and because of clumping, you may not be able to count properly. Remember, we have discussed in the past that a solid staining bacilli with length to width ratio of 1 to 1 is to 3, 1 is to 5 is an uniformly stained bacilli, rod shaped, are the viable bacilli. Otherwise, the fragmented bacilli, the broken bacilli, may not be viable and cause the disease. So what we are trying to say is you take the sample from mouse foot pad, you crush it multiple times, mix it with sterile water, make a smear out of it, stain with AFB, and individually count the solid staining bacilli. Clear? So that is known as the AFB staining. Other method is RLEP-PCR. RLEP is a part of the genome of M. leprae and has multiple copies. You can analyze using PCR that if the copies are present or not, that means if the genome is active or not. This makes the RLEP-PCR very sensitive. So this is more sensitive. So much so sensitive that even less than 300 bacilli per ml of tissue sample, it can still detect. This is easier because computer does it and accurate because you don't have any human intervention. Let's move forward. The fourth application is drug testing. For drug testing, the mouse foot pad assay is the gold standard. Okay, and you require for looking at clofazimine resistance. Why? We know that some genetic mutations can cause resistance against dapsone and rifampicin in M. leprae. But in clofazimine, we are not clear about its mechanism of action. We don't have a good genetic candidate for resistance. So if the genetic candidate is not available and you cannot look at the mutations in specific genes, you need to have an animal model to study the resistance. So for clofazimine, when you don't know the gene, when you don't know the mechanism of action properly, you require an animal model. So mouse footpad assay is the gold standard for drug testing. Remember this, it's a good question or a good viva question. What is the gold standard for drug testing? That is mouse footpad assay. There are few methods to do drug testing. One is the continuous method. In continuous method, the mouse footpad is inoculated by the M. leprae with low numbers. Okay, and then drugs are given on the day of inoculation or the next day, and then you see whether the M. leprae growth was suppressed or not. So if you have a mouse footpad inoculation and a lag phase, log phase, and a stationary phase, you give drugs at day 0, day 1. Because of the drug, if the drug is effective and there is no resistance, there will not be much multiplication. So with the drug, you will not see multiplication and the drug is effective and there is no resistance. If there is a resistance, it will mimic the normal growth. Clear? Another method is the proportional bactericidal group, or the method in which you have immunocompetent mice and you inoculate with serial dilutions. Okay, and you start the drug at day 0 and day 1 up to 60 doses and see that at what dilution the drug is able to stop the growth of bacilli. Clear? So these are two methods. Just remember the name. Continuous method, in which you give the drug to a single cohort of mice, while in proportional bactericidal method you have multiple cohorts with serial dilution of bacilli, and then you give drugs to each of them and see which concentration is limited by the drug. If the drug is able to limit a large concentration, it is effective. If it can only limit very small concentration, it is not that effective and there is resistance. Clear? Let's move forward. The third method is kinetic method. So the mouse footpad are inoculated as in the continuous method. Drugs are started after day 60 when the M. leprae is in its rapidly dividing logarithmic growth phase. So you have the lag phase, log phase, and stationary phase. And here the drug is given in the logarithmic phase after 60 days has passed. Clear? And then you see that what is the lag period between normal growth and after drug growth. So let's say drug is given and then you have the normal growth. Let me just make it again. So when the drug is given, it takes a bit of time for it to reach stationary phase, and this lag phase is measured and see what is the lag phase and tells you about whether the drug is effective or not. Using the kinetic method, you can distinguish between bacteriostatic and bactericidal drugs. Okay, in the modified kinetic method, what you do is you just look at the biochemical test, not the number of bacilli which are restricted by the drugs. You look at the biochemical tests like pyrimidine uptake or oxidized palmitic acid uptake. You look at the biochemical test and to find out whether the growth of M. leprae is restricted or not. And by using biochemical tests, you can do analysis early. You need not wait for 60 days. You can do it as early as 30 days in a modified kinetic method. Clear? If there's any confusion in the methods, just let me know in the comment section. I will explain again. You just remember the names: continuous, proportional method, kinetic method, modified kinetic method. Clear? Let's move forward. Now, how to evaluate for vaccine candidates? The process is nearly the same as using for drugs. Instead of drug, you just give vaccines. So Charles Shepard was the first person to use mouse footpad for vaccine testing. Mouse footpad is the animal model choice for vaccine candidates. You give vaccination either intradermal or subcutaneous plus a low dose, 5,000, roughly about 5,000 bacilli per footpad of inoculation. And then you harvest the tissue at about 6 months after the challenge. So what we are trying to say that initially you give vaccine plus inoculation, and after about 6 months, you take the sample and see whether M. leprae is living or not. We see the percentage or fold inhibition of M. leprae growth. That means we have two different mice. You give vaccine to one mice. You do not give vaccine to other mice, which is the placebo. You inoculate both footpads and see if there is a proportional decrease in the growth of M. leprae in the vaccinated mice. And that is how you check vaccine candidates in mouse footpad method. Heat killed M. leprae 10 raised to power 7 in number is used as a positive control. Let's move forward. Limitations of mouse models. Now you need to mention, whenever writing a short note, you need to mention a bit about limitation of mice models. Now mice models, they lack a homologue for granulysin. Granulysin is an enzyme which is secreted by natural killer cells and CD8 positive cells, and they're responsible for killing of M. leprae, for destroying M. leprae. Mice don't have another protein which has the same function as granulysin, so you cannot study that. The CD1 cell surface markers are different in mice as compared to humans. We require CD1 cell markers to identify lipid antigens. Clear? Mice models have more robust nitric oxide synthase to generate nitrogen intermediates in mice models. So you have more nitrogen oxidation damage as compared to humans. They recognize different antimicrobial mycobacterial responses. Mice are highly resistant to infection with M. leprae unless they are immunocompromised. We cannot have good spread of infection in animals in mice models because they are inherently resistant to some extent against M. leprae infection. That is, and also they are not ideal for neural infection. Remember, nerve tissue of mice resist M. leprae infection, so you cannot study properly nerve involvement. Clear? Let's move forward. Okay, so now we have studied a bit about mice models in leprosy. Let's come to the animal model that we use routinely now, the armadillo model. Armadillos which are used are the nine-banded armadillo, scientific name Dasypus novem cinctus. Remember this, this is important. In Viva, it is, I've been asked what is the scientific name of the nine-banded armadillo. It is Dasypus novem cinctus. It's a good MCQ question also, just remember that. Armadillo is the only other natural host of M. leprae. That means the only organism except humans which is infected by M. leprae. So much so that they have been implicated in zoonotic transmission, that means transmission from animals to humans. So it has been postulated, not postulated, it has been actually proven that M. leprae can get transmitted from armadillos to humans also. They are host of choice for propagating large quantities of M. leprae. Remember, in mice, you cannot use large inoculation or the mice will get immunized. The immunity will destroy M. leprae. But in armadillo, large volumes of M. leprae can easily be inoculated and can be cultivated. Also, neural infection can be studied because the nerve involvement is similar to what we see in humans. So different from mice models, you can actually study nerve involvement in armadillos. Let's move forward. In armadillo, the entire spectrum is seen from tuberculoid to lepromatospore. You need not use an immunocompromised, immunocompetent. Armadillo is good enough. About 70% of armadillos show an LL response. About 15 to 20% resist infection. Like in humans also, not everybody develops a profound leprosy disease. Some of them can harbor bacillus without developing the disease their entire life. Siblings and mothers behave the same. What do we mean by that? Now, what happens in armadillos is that one mother will give rise to about four armadillos. And the four armadillos are somewhat of a monozygotic organism. That means they share the genome. And these four siblings plus the mother will behave same. That means if the mother is developing LL Hansen, then the four siblings will also develop LL Hansen. Clear? That's what we mean to say. The nerves are affected, they have functional deficit and the nerve inflammation. Manifestation depends on the particular animal's immunity. That means, like I said, if the mother is developing LL Hansen, the siblings will also develop LL Hansen. If they develop tuberculoid pole, the children will also develop tuberculoid pole. The standard challenge dose is 10 to the power 9 M. leprae. So you can use a higher inoculation as compared to mice models. And it takes about 18 to 24 weeks for the lesion to properly develop and harvest the tissue. Let's move forward. Now, this table is just in comparison of mouse models and armadillo models. We know that mouse are easily available, so availability is good in mouse models. They have ease of handling, small animals rapidly breeding, so we can handle them easily. They are small size, you can easily keep them in enclosures, and they are readily available for research. While armadillos are large size, they are roughly the size of house cats, cats that you have as pets. But advantage is that they have a natural susceptibility to infection by leprosy. Remember, mice models can have natural resistance to development of leprosy infection. Well, armadillos have a natural tendency to develop similar leprosy that we see in humans. You require a large lab for armadillos because the animal is large and you have to procure them from Americas. You cannot breed them in captivity, so you need to get them in a wild form. Other non-human primates like chimpanzees and mangrove monkeys, they're used for long-term study. Best is used is long-term study because incubation period is about 20 to 24 months. So mouse model is the gold standard. It is most convenient and reliable. In fact, it was used to design the WHO recommended multi-drug treatment initially. Remember, in about 1983, the MDT treatment was started. So roughly, if I remember correctly, so mouse model was used because you need to do research quickly. But now armadillo animal model is used more. So with that, I'll finish the lecture. We have multiple different lectures of different aspects of leprosy already on the channel. We will have newer lectures in the coming weeks. We have discussed drugs in leprosy, alternative therapies in leprosy. The lectures on immunology in leprosy is, in my opinion, it's a good lecture. It took me a lot of time to research and develop these two lectures, and it will clear your concepts about what actually happens when leprosy enters the body and what the body does to M. leprae when it encounters it. The lecture on pathomechanism nerve damage is important theoretical wise, and biology of M. leprae tells you what organism you're dealing with. The Zeal-Nielsen staining and slit skin smear are good lectures, and I've gotten good response after uploading those lectures. So these lectures are available as of now. Newer lectures will be coming in, continuously being added in subsequent few weeks. So search for these lectures on the channel and look at them. I would highly recommend Immunology of Leprosy and Pathomechanism of Nerve Damage. These are good lectures. Let's move forward. So with that, these are the further reading recommendation. This is a chapter in IAL. It's a good chapter, but multiple paragraphs are not required at residency level. You just need to have a basic understanding. What are mouse models? Why are they used? What is the inoculation size? Mouse foot pad is a gold standard. Why are the armadillos used? What are the advantages? What are the limitations? What nine-banded armadillo? Okay, you need to know some aspects so that you can write a good short note when a question comes on animal research in leprosy. I would recommend that you read the Rees article and you read this article from infection to biological model. These are good articles if you have time. Or you can just go through this lecture and that is enough. You will have good understanding of how animals were used to study leprosy. Okay. Again, requesting you to become a member. If possible, the least minimum level of membership is Ruby membership. You will get access to longer videos which have bonus contents, extra information, and becoming a Ruby member motivates me to create more and more videos. And if possible, if you think these videos are helpful for your residency or post-residency dermatology training and understanding and learning, consider becoming a Sapphire member. Okay, with Sapphire member, you have earlier access to videos. It's a level above Ruby membership, and in fact, I keep on posting polls to decide what next topic should be used. In fact, the segment to study, which is leprosy, which will be studying a few topics from leprosy for the subsequent 3-4 weeks, were decided by the Sapphire members. So, with that in mind, if possible, do consider becoming a Sapphire member. Okay, with that, the lecture comes to an end. It was a long lecture, but only this lecture would be sufficient enough at residency or post residency level to have a good holistic understanding about animal research in leprosy. Okay, you will have enough information to write anything, answer all your vivas, and to have a basic concept that what actually happens when we say that we are using mice models or armadillo models to study leprosy. Clear? So without much further ado, if you have any doubts and queries, you can email me, or it's better to comment with using a timestamp in the comment section. Without much further ado, I will close this lecture. Adios and bye bye.